Okay, I think the recording has started.
Yeah.
So, hello everyone. For today, we have like a excellent guest, Gabriel, that I have met 1 year ago, and I'm really excited to hear about like his work. He has been kind of revolutionary, like for the field, like doing this kind of new experiments, and I hope you all enjoy. And for the webinar, we are going to restart next year. I'm a bit unsure when exactly, if it's going to be February or March. But if you are interested about sending your suggestions, please feel free. And without more delays, please, Gabriel, the floor is yours. Thanks so much for coming.
Yeah.
It's a real pleasure.
Thanks, Lucas. Yeah, thanks for inviting me. I've been invited to present this work that we've published about a year ago about inducing novel endosymbiosis by implanting bacteria and fungi. And I'll try to keep the introduction quite short of why we're doing this. Basically, we are interested about endosymbiogenesis in the very early stages. So rather than looking at established system of different ages, we really try to experimentally probe what happens when, in our case, usually endosymbiotic bacteria start moving into a host. And maybe one distinction that is not always quite clear with endosymbiosis is from the membrane topology of how this works. And several things can happen. Some of them aren't like properly intracellular, but we're really thinking about intracellular endosymbiosis where the bacterium is fully engulfed by the host cell. And in this specific case we're presenting, it's also directly in the cytosol and not actually surrounded by another host-derived membrane. So it's not in a vacuole, but directly in the cytosol. And so we've thought about this and we've done some experiments as well. And this is a quite challenging process for an endosymbiont to make this work. If you just pair a random bacterium with a random host, chances for this working are basically zero. And that's why we've started to set out to probe this more experimentally with a natural system that has quite some pre-adaptations to the endosymbiotic lifestyle, where we thought we can get some interaction to work, and then we can learn about what these organisms have to overcome and to adapt to become even better at endosymbiosis, to show us where really the optimization is necessary to lead to stable endosymbiosis.
Uh,
And for this, we work
can I make
with
a
this small
small
system.
question?
Yes.
So I was wondering, like, why bacteria as a guest? Like, I don't know, like, would it be a unicellular eukaryote? Did did you have any specific reason to starting with bacteria? Maybe I
Um,
suppose it's simpler, but
yes. I mean, and so biotechnologically, we will be quite interested in bacteria. I think they're a great model platform. I mean, so there's two aspects. We would like to learn, of course, about the evolutionary forces behind this, and also to understand evolutionary history a bit better. But we're also seeing kind of biotechnological applications for this in the future. So that's where bacteria coming quite handy, we think. And then there's also a size issue. So for the technology we had in mind to use for this, or that we use for this, you need to be able to really inject the bacterium into the host, and the larger the hole you need to make, the less likely the host is to survive. And then, of course, archaea would also be interesting, especially for evolutionary history. And there it's mostly about how to culture them and how to work with them. That makes this extremely tricky. And also the choice. So we see that a lot of endosymbiosis that fascinate us in the lab are bacterial endosymbionts. Is why we kind of focus on this.
and why a host being a fungi?
Um, uh, there's several things, so we had to start with something. We do again think fungi are biotechnologically relevant. We've also done work on like HeLa cells, but for example, in mammalian cells, you run into a lot of complications, such as the immune system that the cell has, that we think is just a lot reduced in fungi. Um, and yeah, so they're biotechnologically relevant. And there's a lot of interactions. So we know that some of them harbor endosymbionts. Many of them actually harbor endosymbionts. And if you look at soil, it's chock full of of bacteria and fungi. So it's an important association. But we wouldn't, I would never say that fungi are the only interesting thing. I
No,
mean,
no,
I love fungi, but we could definitely work with other host organisms and some people in the lab even, we're trying to expand this. But every system comes with complications.
very cool. Thanks.
Yeah. But so for this system, we started with the Rhizopus microspores, which is a fungus. It's a somewhat special fungus because it doesn't form regular septa. So it's so an acidic fungus. The compartment is much larger. And what was really cool about this, what allowed us to do these experiments, is that there are some strains of Rhizopus microspores that harbor Myceta habitans rhizoxinica. So it's an established endosymbiosis that is not completely obligate, but relatively well synchronized. So, for example, the bacterium's genome has been reduced by about half, but it can still be cultured in the lab. So it still retained a lot of kind of like the metabolic abilities that it needs. And so I brought a short video. I hope you can see this. It also kind of shows you how well synchronized these two organisms are, because the bacteria here are labeled in green. They really can actively be transported through this mycelium. So they really form a good team, right, in the natural state. And like I said, so this is true for host strains. It's relatively recent if you look at a very reduced endosymbiosis, but it's a team effort. The fungus is quite special because it has this complicated life cycle. It usually forms a large mycelium, but then it has to do the spore stage to disperse far. And in these spores, the bacteria are really reliably inherited. So almost all of the spores have the bacteria. And to make sure that kind of this endosymbiosis is stable over many generations, there's also this kind of safety switch that if a spore doesn't have a bacterium and then you have an empty mycelium, or in the lab, we can do this with antibiotics. If you take the bacteria out of the host strain, then the host strain is still viable. So it's not per se obligate, but it will not form any asexual spores anymore. So it's kind of cut out of, its future.
That's very cool. Do you think the bacteria is inducing the fungus to the?
Yes, so we know that in these host strain, the bacterium is necessary to induce the sporulation, and there's quite some research also trying to figure out how they do this. I think the best indication we have is maybe the type 3 secretion system, where they have like transcription activator like proteins that they secrete that seem to do something in the fungus to activate the sporulation. But again, so this is what we could work with, is that there's also non-host strains. And these non-host strains, they naturally do not harbor any endosymbionts. And that means that also like they have the same life cycle, but the sporulation is not tied to harboring endosymbionts. And these non-host strains are actually known to rather defend themselves against Myceta habitans if you challenge them in the lab with the bacteria. So they mount a defense reaction against it, and they cannot be naturally infected with the bacterium. And we thought, well, this is a great way for us to study what happens if we have a bacterium that does have the fundamental abilities to live as an endosymbiont, and inject it into an organism that doesn't naturally harbor them. And so where it shouldn't form a stable system from the get-go, but how they adapt. And then, so the goal was to inject bacteria into a non-host strain, see microscopically and live whether these two organisms are then both viable, or if they start killing each other, which is a possibility, right? And then we would also see how stably this can be passed on to further generations of the fungus. And then, and especially the spore stage, that is quite tricky to navigate for an endosymbiont. And so, because here everyone is aware about endosymbiosis, right? But forcing this uptake is quite challenging, and it was quite new for us to be able to do this. And this was centered around this technology, which is called the Fluid FM. And I'll take some time here to explain it, just so that people maybe know what we're working with in our lab. The fluid FM is this basically an atomic force microscope, a small, modular, easy to handle atomic force microscope. And then you can mount this on basically any inverted microscope you want. In our case, we have a confocal spinning disk microscope associated with it as well. And the special thing about this AFM that makes it a fluid FM is that you can actually mount cantilevers with which you would tap the surface in a traditional AFM that is actually hollow. And you can use this as a very, very small pipette by applying under or over pressure to the system. And this comes off, and you can use different kinds of probes for different kinds of applications. And then if you mount your AFM onto the microscope and you put your sample inside a Petri dish, then the Petri dish goes in first, and then the AFM. And what you end up with is your sample at the bottom, and then the ability to approach and inject or extract from your biological samples. This technology comes with its own difficulties, but it allows us to see in real time the manipulations we're doing, and we can be relatively gentle with the cells that we're treating. That doesn't mean it's always extremely gentle, but we can have relatively high viability of different cell types. Now, when I started my PhD or this project, I did have to do some method development where the fluid FM wasn't quite ready to handle this. This has mostly to do with using fungi rather than mammalian cells that the lab had used before, because they're thin, they're hard, and they're under pressure. So then we had to kind of like find how to make even sharper tips to be able to penetrate the cell wall. We use some enzymes to soften the fungal cell wall a little bit. That also makes them swell and have a little bit more volume to it. And then we had to kind of tweak the maximum forces and the maximum pressure that we can apply to overcome this hard cell wall and the high internal pressure of the fungi.
I have a question.
Yes.
Do you think it's possible to do the same with plants?
Yes. It's plants come with their own kind of like difficulties, and you couldn't just use a leaf or something. But in cell culture, yes. So we're kind of looking into this, yeah. But again, so technologically, it's just if you really change the fundamental like physical characteristics of a host that you're using, then you need to do a lot more method development than if you're just changing the bacterium, right? The bacterium is mostly defined by its size. There can be other issues like stickiness and stuff. But this is relatively easy to exchange, and then the host, you just do optimization. Yeah. And so because we could put this together, I can show you this video of how this worked with the injection, where we now inject the mycetohabitants into the non-host Rhizopus microspores. So what you see here is a germling. So this was one spore of the fungus that started to grow and is starting to form kind of like the hyphae that will eventually form a mycelium. And in black, you can see the fluid FM cantilever that is hollow and we're inside the green labeled bacteria. And I'll let this play. So here it's actually, this is the exact position where we're injecting. And you can see now we're inside, there's backflow into the probe because there's a pressure in the fungus. And now we start applying overpressure. And all of a sudden you can see now liquid is actually flowing in. And because the bacteria are labeled, we can then see that the bacteria are really starting to flow into this germline. And then after we use this overpressure, we have to reduce the pressure again to let the germline recover. It has to close the hole. And then we change to time-lapse imaging of the bacteria that are now injected in this fungus. And then we can see how they are starting to move and grow. And you can see for sure here the fungus has survived this procedure. It survives having bacteria inside, and it starts growing really, really rapidly. That was already been quite exciting. I mean, it did take quite some optimization. And if you look closer, which might not have been obvious from the video, is that you can also see that the bacteria also managed to divide. And this is not obvious. We know that a lot of other bacteria will actually not be able to survive in the cytosol of a fungus or any other host. But in this case, we have metabolic compatibility, right?
Do do you have any intuition why they would not be able to survive?
Um, I mean, so there's an easy answer to this, which might not be so satisfactory, and that's just saying, well, if you just get a random bacterium and try to grow it in the lab, you'll probably have some hard times, right? Um, Most bacteria just have relatively specific nutrient requirements, and even if you try to give them something really rich, and we assume the cytosol is quite rich, like Lb, a lot of bacteria will refuse to grow on this, right? Um, and then, there's some other factors that will definitely play a role. The cytosol is a living compartment, so it is extremely regulated. It is very different if you look at the nutrients. If you give a medium in a shake flask, you just give them a big bolus, excess amount of nutrients. In the cytosol, there's never this big bulk of these nutrients available. There's usually a very well managed amount of all the chemicals or a lot of the chemicals in a cytosol, where then you will have to think about fluxes. And while it's quite rich in, carbon sources, nitrogen, like these fundamental nutrients that an organism needs, it can also be quite restricted in specific things. So, for example, like iron, we know is sequestered usually in the cytosol. It might not be so easily used by an endosymbiont. Yeah, so generally biology and then the high regulation of the cytosol. And of course, there can also be active inhibition of growth of the bacteria. Depending on what host, there could be reactive oxygen species that are used to defend against themselves. They can try to be digested, like active defense responses as well. Well, yes. So after the injection, where we saw that both organisms managed to survive this procedure, we were again interested about whether we can vertically transmit this endosymbiosis, which is a really, really big step that is not so straightforward. And in this case, it was complicated because we cannot image the whole mycelium under the microscope. It's on the centimeter scale, and we need like bacterial resolution. So this is not possible, but what we could do is we could harvest the spores, and then we could use a cell sorter that looks for the bacterial signal from the GFP in these spores, and it could check 20,000 spores per second for us, and then also sort the spores that had the bacterial signal into a vial for us to look. And when we did this after the injection, we did indeed find a very, very small subset of spores that had bacteria inside. And, this was just a signal from the machine. I didn't trust it immediately, but I looked at it under the microscope, and I saw that these spores really had the bacterial signal inside. And I compared this to how this looks like in the host system. It did seem to be the real thing. And then, of course, I also took these positive spores that I found, put them in rich medium again. And then I did find some germlings of this non-host in the next generation with the bacterium still inside, and actually better density of bacteria than after injection, which just means it's just because they have been in there from the beginning. But what I also saw was that the fungus formed way more spores that didn't have bacteria, and the bacteria containing spores were relatively bad at germinating. So I had to seed a lot of them into rich medium to get just a few germlings out. So that told me this system has vertical transmission, but it has a very low fitness. It has a high fitness cost basically on the fungus, right? But vertical transmission means you can do adaptive laboratory evolution, which is a very, very powerful system, right? Where we let biology kind of like figure out a better way to cooperate by themselves, as long as we have a very strong selection pressure. And what we did is we took these
Can
positive
I
spores.
can I ask a question?
Yes.
Um, why do you know what's the reason why not all the spores go to the bacteria? Is because they're not reproducing like in sync? Would that be the reason, or?
Yeah. I mean, it's multifactorial. One of them is that the density of the bacteria seems to be lower in the non-host than it is like in the native system, right? So there's not as many. Another thing is, so you see in the native system, almost all the spores have bacteria, but this is because there's an orchestrated process going on, most likely, that says, well, spores are only formed when bacteria are present. And then there's likely some sort of checkpoint where the spore will not kind of like ripen and close off until it gets the signal that the bacterium is inside. And the non-host doesn't have this. The non-host just tries to make its own spores. And whether a bacterium ends up inside it or not is rather an accident. If like it's a bacterium, it doesn't really have a will, but as a human, if you think about it, we can say like the bacteria maybe want to end up in the spores because it's their chance for the next generation. So they might try to sneak in a way, but the fungus doesn't wait for them. Yeah. And so to the adaptive laboratory evolution experiment, so we took 10 lines and then we kept this going for several rounds of always strongly selecting only for the spores that have bacteria. They then select for the ones that survive, and then do this over multiple rounds. So after 7 rounds, we narrowed it down to the best performing lines as well as a pool, and then only had 4 lines going for 10 rounds. And what we saw was that during this adaptive laboratory evolution, the fitness of the pairing increased dramatically. And so the fitness, we here describe this as the percent of viable offspring with endosymbiont, right? So from all the spores that are formed, how many of them have an endosymbiont and can germinate? And this is made up of the germination success of the spores that have the bacteria inside, which in the beginning was really low, but then came up to levels that were similar to what spores have that do not harbor any bacteria. And it was also made up of the amount of spores that get a bacterium inside. And this positive fraction also increased. So in the beginning, this was extremely low. And after the selection in the best performing lines, we actually had a positive fraction of almost 30% of spores that had the bacteria inside. And if you multiply these two values for each of the data points, then you get kind of this fitness index that, of course, increased dramatically over these 10 rounds. Again, here we have kind of a selection of the best performing lines that we did artificially. And of course, as biologists, we want to know what are the underlying genetic factors that are the reason for this. And for this, we did population sequencing, both of the fungus as well as the bacteria. And we did this at the kind of the endpoints at around 10, but also where we had all the lines going. And the best performing line, which was line 4, we also have kind of time resolved data to see when did what change. And when we looked at and mutations that started enriching in these populations, we fortunately found some mutations that made the threshold. And we saw also that the best performing line 4 actually had relatively many mutations. What was quite interesting was that we found the mutations only in the fungal population and not the bacterial population. Intuitively, we would have thought kind of that the bacterium mutates faster than the fungus, but this seems not to have been the case here. We saw that the genetic changes we saw in this time resolved line 4 were highly correlated with the fitness gain, as in at the same time that the fitness of the population was increasing, so more spores were formed with bacteria and they germinated better. These mutations that were found already early on in the experiment started increasing. So in the dotted line, you have the fitness of this line 4, and then you have these 4 different mutations plotted over time as well. And you see that they are almost fixed in the fungal population at the end. However, this is really a correlation, and to this day, we cannot say that these mutations are the underlying cause for this. We do not have proper proof of causation. And we also don't know exactly about the function at the molecular level that these mutations had, because they're most likely having something to do with transcriptional regulation and the like, where it's kind of broader changes and it's not just single enzyme that started changing. So we don't know exactly what was necessary to increase the fitness of the system from the host side. But we do have phenotypic indications of what kind of functions in the life cycle of the fungus changed to adapt to this endosymbiotic lifestyle. And there was the main clue was actually that we saw in the FACS data. So when we went to the cell sorter and analyzed how many spores had bacteria inside, where we saw that the positive population decreased in intensity. So like over time, we had more positive spores, but they were kind of like less bright. And then we figured, well, this less bright mean that they have fewer bacteria inside. And so we did this by analyzing 3D image, like set stacks of the spores of these spores. And we saw that, yes, this decreased intensity correlated to a lower volume of bacteria inside the spores. And when we looked at the fitness influence that this had on the spores, this correlated quite well. So it seemed that if a spore had a lower amount of bacteria inside, it was better able to germinate. So in the beginning of the evolution, there were already some spores that had, less bacteria than other spores. And these already in the beginning were actually better at germinating than the ones that had a lot. At the end of the evolution, all of the spores had less bacteria inside. So even the ones there that had the most were still rather similar to the ones that had the least in the beginning. And then they all also started to germinate much better. So this seems to have something to do, which would make sense if the bacteria use too many resources of the spores. So we think somehow these mutations are involved with the fungus reducing or keeping in check the bacterial population in the spores, but we don't know how on a molecular level this works. Now, we are in the lab, we're mostly interested in mutualistic endosymbiosis and not just like studying pathogenicity of bacteria and fungi. So we're also interested in the potential benefits that this endosymbiosis can have on the host. And for this, we looked at also again the native system where we know that the bacterium supplies the host with rhizoxin, which is used to defend itself against predators, but also it can be used to find nutrients by attacking plant roots with it. And we wanted to see whether now the non-host that we've checked cannot produce rhizoxin because it doesn't have the endosymbionts, now has this rhizoxin available when the induced endosymbiosis is going on. And indeed, we found both, like in all the 10 different lines, we found rhizoxin itself as well as a precursor, so a small interesting fact about this is that the bacterium actually produces this precursor, which is then modified by enzymes of the fungus to produce the final product rhizoxin, which seems to be also the case in this non-host strain. And so this was really exciting because it didn't only mean that we induced a new endosymbiosis, but also that we transferred a metabolic trait with the endosymbiont, right? But like looking into the future, we would also like to maybe transfer other metabolic traits that aren't inherently possible for this bacterium that we chose. So we would like to go to a more platform organism like E. coli potentially, which is a lab strain. Everyone likes it. Many things are possible with E. coli. And we wanted to see, well, what happens if we inject E. coli?
Can I ask a question? So for the find that you just show, did you try to see if the fungi could use that new metabolite that was being now produced that effectively, I don't know, like killing or whatever is in the environment or trying to access the root?
Yeah. No, so we've struggled. I mean, we didn't spend too much time on it, but we've tried to do like a biological assay with it, and we couldn't really see the effect of the rhizoxin. It's in the lab setting, it's always hard to say like what kind of concentrations are necessary. I do think there were significant amounts of rhizoxin present. So if we ran standard, like if we ran comparisons with the native system, we weren't like orders of magnitudes off with the amount of rhizoxin in it. So whatever naturally the benefit is of rhizoxin, we think there's a good indication that this could also be the case with a non-host, but we didn't show this in a biological setting.
Thanks.
Yeah. So we tried with E. coli. It would be super nice if you got a stable endosymbiosis out of it right away. But the dynamics were quite different when we injected E. coli rather than mycedohabitans. What you can see here is from I didn't bring the video, this is just like a few snippets out of it, is that after we injected E. coli also managed to divide in the in the rhizopus. So it also has this metabolic compatibility. And what was also striking, I mean, this was really nice for us, is that the fungus indeed also still managed to grow with E. coli present because it grows quite fast. So it didn't like immediately die because there was E. coli inside. But what did happen was that E. coli divides much faster than mycedohabitans, and it isn't as mobile within the mycelium. So it doesn't disperse as effectively. And that meant that locally E. coli started growing to extremely high densities. And at these really high densities, it did have kind of a negative effect on the fungus, and it started stressing the fungus. And then the fungus reacted to this stress, and it started building septa. So it made walls in its hyphae that it doesn't usually have. And it wasn't great at capturing the E. coli. It just kind of made it in the areas where E. coli was too dense, which meant that there were areas where there was just still a few E. coli that kind of managed to escape that kept this going for quite a while. So the E. coli could still colonize relatively long stretches of hyphae, but it meant that there were some areas where it started to completely kill the fungus. So in this area where it's all E. coli, the fungus doesn't survive. But there were also areas of the fungus that were completely bacteria-free, and these grew super happily. And that meant that when we then checked for, metabolic compatibility, when we tried to check for the spores, whether we found spores with E. coli inside of them, we couldn't find them. There wasn't a vertical transmission because they were confined too early. And then these areas that had the bacteria weren't really fit viable anymore. So there was a counter selection against this endosymbiosis before it could even get to the stage of vertical transmission. Yeah, so these are already the main findings of the paper that we published about a year ago. If you haven't seen, about a month ago, a postdoc in our lab, Tom Gosler, he also made a really nice study where he used the same injection technology, but now used Ralstonia as an endosymbiont, which had a much more kind of pathogenic effect, like E. coli, but still managed to be vertically transmitted. And then we could do transcriptional studies during an adaptive evolutionary experiment where also the fitness slightly increased, but just never got as good as with mycedohabitans. But we could see how the fungus started to go from this antagonistic response against the bacteria and kind of become more mutualistic towards it, so more neutral towards it. It started decreasing kind of these antibacterial responses that it had over time. So if you're interested in this, you can read up more on this study. I didn't have time to present this finding in detail as well.
Can I have a small
Yeah,
question before I take
please.
home? In that cases that you saw the E. coli being very taxing to the host, did you find any case in which the bacteria would grow so much that you would explode the host? Or
Yes, they can actually explode. Yeah, this is actually kind of fun. I even have videos. I don't think I can show it right now because I don't know where to find it. But yeah, so these compartments where it's kind of like a small compartment, it's full of E. coli. The E. coli at some point, so I assume they start to feed directly from the medium because the fungus dies. Then the membrane becomes leaky and nutrients from the medium outside come in. and the E. coli just keep growing, and they can put up so much pressure, which so the fungus withstands about 4 or 5 bars of pressure quite easily. They build enough pressure to really burst these compartments. So yeah, we could actually see this kind of happening live. Yeah. So yeah, as the take-home messages, I hope I could show you that in our lab with the new adaptations of the fluid FM, we could explore some really interesting new opportunities. And by implanting bacteria, we enable the induction of a novel endosymbiosis that doesn't exist like this in nature, and see in real time kind of what happens, and then use adaptive laboratory evolution to stabilize this. And in our case, this came with a large host influence, but of course, this might be different if you use a different pairing. And also what's quite exciting for us, also for the future, is that with the endosymbiont, we could actually transplant the metabolic capabilities of the bacterium to the new host, which also, of course, evolutionarily makes a lot of sense because this is kind of the big gain that you can get from undergoing endosymbiosis. And in the future, you might be able to use this for biotechnological applications. Unfortunately, what we saw was that bacteria with lower levels of pre-adaptation aren't vertically transmitted. So we will still have to learn a lot more about what is really necessary, like what are the abilities that mycetohabitans has that E. coli doesn't have, to start engineer this in all these bacteria that we're interested in as potential endosymbionts. And it also shows you that how in evolutionary history, it's been really a big step for a bacterium to become an endosymbiont, right? This is not super easy to happen. It actually takes quite a lot of prerequisites, and still we see it popping up, across all the branches of the tree of life, just because there's so much to be gained from a successful endosymbiosis as well. Yeah, so I would like to really say thank you to so many people involved. A big shout out to Julia, my PI, who allowed me to do this research. And then we have a lot of very talented people in the subgroup, former and present, and also a few that have just joined recently that are really driving this research further. And then, of course, this was only possible with international collaborators as well as collaborators from inside the institute that helped, and the facilities, which was quite special for us because, for example, the fax we were really happy that they had the courage to let us sort fungal spores in their machines. So yeah, thanks a lot to these people. And yeah, if you have more questions, I'd be more than happy to answer them.
No, thanks so much. It was like excellent. I really had fun to listen to your presentation,
Thanks.
and it's much more digestible, like hearing you talking about than reading it. So
Yeah. I mean, the papers have to be dense, right?
yes. Well, I think I had all of my questions, except one. I just remembered, like, so do you think part of the E. coli being so deleterious or bad for the host is big? Does it have like a higher, longer or bigger genome than the other one that was beneficial? I don't know if like maybe because it has a bigger genome, can do more stuff, so there's more likely to cause harm. I don't know if that is.
Yes. I mean, I wouldn't necessarily just say it's just because of the bigger genome size. I'm sure there would be other bacteria that have even larger genomes than E. coli, that just wouldn't be able to grow inside and then wouldn't cause any harm. Or some that have large genomes and still tend to grow relatively slowly, even in rich circumstances. But yes, kind of like this too fast growth, like the metabolic abilities that E. coli has to really make use of a lot of the nutrients in the fungus. I would assume that this has to do with this deleterious effect. And we do think it would be very helpful to slow E. coli down. And yeah, I mean, like the degradation of the mycetohabitans genome, like, or that has become smaller, and we know that it has lost, metabolic capabilities. There's an assumption that it mostly grows off C3 carbon sources actually, and ignores C6 carbon sources. And E. coli, of course, would also eat all the C6 carbon sources, and many other things it finds might help to synchronize the host and the endosymbiont, right? So that you would want a stable endosymbiont to kind of sense the energetic state of the host, and to adapt, and not to just being able to grow in the host. It isn't really enough to be a good endosymbiont. Yeah.
Well, I think that's all. Thanks so much for coming. I really enjoy
Yeah.
chatting with you and for the awesome presentation. And
Yeah, it's been really nice um, meeting up with you again. I don't know, are you going to the EMBO in Heidelberg next year?
I'm not so sure yet, but
Okay.
I would like to, but I have to see. Yeah, maybe I
Yeah.
can stop the recording just a second.